First of all, good morning. It is a pleasure to have this opportunity to talk to you about our experience using the xCELLigence system in biocompatibility testing and also in assay quality control in the medical device industry. As Lena mentioned, I am a cell and tissue culture biologist at KCI, which is a wound healing company headquartered in San Antonio. And today, what I'd like to do is talk to you about three things. One, really briefly about what KCI is all about and why we love working for our company. Then I'm going to tell you briefly about our negative pressure wound therapy system and how we use it in vitro on primary cells. And of course, I'm going to devote the majority of the talk on some experiments we've done in the laboratory, proliferation and migration assays using the xCELLigence, which is really why we've all tuned in this morning. So typically, we show some pretty gory pictures of real wounds that you might typically see on our patients. And the reason we usually use real pictures to depict these wounds is because most of the time that is really what's going to drive home the message that these wounds are really detrimental to the quality of life of a lot of our patients and a lot of our loved ones. But because we're doing this so early in the morning today, I decided to spare you from some of those pictures. And instead, I'm showing you on the left a schematic diagram of a foot ulcer. This foot ulcer on a healthy patient would rapidly progress through the three major stages of wound healing, which of course are inflammation. Then, of course, we have the proliferation phase, where different cell types are recruited to the wound and collagen builds up, allowing for full closure. And the final stage, which is remodeling, which can sometimes take up to several months to a year in a healthy patient. Unfortunately, unhealthy patients tend to get stuck in the inflammatory stage and can have an open, gaping wound prone to infection for many, many years. And so right now, millions of patients require clinical intervention for some of these types of wounds, and the cost of treatment is more than $20 billion. So one of the focuses of our company is primarily on chronic wounds, on closing hard-to-heal wounds, which unfortunately, as you can see here, is on the rise from 7 to 10% predicted growth rate by 2016. So we focus a lot on these chronic wounds, diabetic ulcers, venous ulcers, and pressure ulcers, try to provide solutions for our patients. So just to give you an idea of how one of our products heals chronic wounds using negative pressure, we call the product the V.A.C for vacuum. And as you can see, first, we affix an open cell foam in direct contact with the wound. We then drape the wound with a semi-permeable adhesive. We apply our negative pressure system, and then the suction is turned on. The therapy promotes healthy granulation tissue formation, which then allows for closure using secondary and primary intention. This is an active suction therapy that brings the wound edges together. It removes infectious material. It reduces swelling, and it induces vasculature. And also, we induce granulation tissue formation. And we do that by recruiting cells to the side of the wound, and also by allowing those cells that are now recruited to the side of the wound to proliferate. And this is really what we focus mostly on in the laboratory. We look at the mechanisms of action for V.A.C at the cellular level. And we're going to talk more about how we employ the xCELLigence in order to accomplish that in just a moment. But first, what I'd like to point out is that there are different cell types that we work on in the laboratory. Mostly all of them are primary cell types and all of them found in the skin. As you can imagine, we work with human epidermal keratinocytes, which are important for epithelialization. We work with human dermal fibroblasts, which are important, of course, for granulation tissue formation. We work with adult mesenchymal stem cells, which are important for just about everything. Endothelial cells, which are important for angiogenesis and vasculature. And we also work with immune cells, which are, of course, important for protecting our skin from infectious materials. So we do work with all these different cell types with the xCELLigence currently in the laboratory. So we focus on two major things. One is efficacy, right? How does the V.A.C therapy work on human primary cells in vitro? How can we improve upon those mechanisms of action? The other thing that we really focus on is safety. Whenever we are designing new products, we want to make sure that whatever new materials we're considering for our products are going to be biocompatible, that they're going to be safe to use on human skin. So I'm going to focus the talk on those two different aspects. First, on how we test some of our mechanisms of action using negative pressure in vitro, and then later a little bit more on how we test biocompatibility using the xCELLigence. So traditionally, what we do for testing mechanisms of action is we culture primary cells, any of the primary cells I just mentioned, for some time. We then encapsulate those cells into a fibrin matrix, and then we actually treat them with negative pressure wound therapy in vitro using a KCI patented system that allows us to, it's a bioreactor that allows us to impart negative pressure quite readily in cells that are being encapsulated in fibrin clots. We then, previous to having the xCELLigence, would then treat our cells with, or we would test the cells either for effects on other cells that have been laid down on a monolayer through a scratch assay or things like that. But I think, as most of you probably are already familiar, the scratch assay is extremely time-consuming, and it's not always reproducible. I think that I'm showing you here just one example of what it looks like. In this assay, we first culture a monolayer of cells, then using the pipette tip, we create a defect in the monolayer of about 1 millimeter thick, and then we monitor cell migration over time. The issue with this method is that we need to monitor the cultures for migration at 2 hours, at 4 hours, at 8 hours, 12 hours, 24 hours, 48, even sometimes longer than that. And because different cells migrate at different rates, and it really doesn't help to compare two treatments if these in-between stages have been already missed. Sometimes this goes from here to here in a matter of hours, and it's really difficult to tell exactly what the kinetics is going to be. So it is really important to really monitor these monolayers quite extensively. And so you can imagine that you spend a lot of time in the microscope, and the hours are really rigorous, and it's very time consuming. This is one method that we use typically for assessing cell migration or in wound healing, in the wound healing field. The other thing that we can typically use is, of course, a traditional migration assay using a Boyden chamber. And I think that those of you who have used a Boyden chamber in the past also are familiar with how time-consuming and really difficult to set up that particular method is. It's worked for a long time in the past, but it really isn't time efficient, and it isn't exactly reproducible either. So with the xCELLigence, time points are a non-issue. The xCELLigence monitors migration at 15-minute intervals, which is really our preferred setting for any length of time. So now we analyze the effects of secreted factors in media after back treatment on different cell types using the xCELLigence RTCA system. And I think most of you are familiar with the technology, but I'd like to briefly review how the xCELLigence technology works so that we all have a good understanding of why it works so well for us in the lab. So the xCELLigence system is, of course, cell-based. The assays are performed in real time, and one of our favorite things is that it is label-free. So we are not introducing any additional chemical compounds to our cell cultures that might impact our results. It consists of the RTCA analyzer. In our case, we use a dual DP analyzer, which fits both an E plate and a SIM plate, cell invasion and migration plate. So we like to do both proliferation and migration assays to study wound healing. The next part of the system is, of course, the RTCA control unit, which sits outside of the incubator on the benchtop right next to the incubator. You can see the RTCA analyzer fits quite well into our incubator. And what I'm not showing you here, but right above this incubator, we have another incubator with 2 xCELLigence DPs sitting side by side. So we have 2 in the upper incubator and 1 in the bottom incubator, just because we really use it quite regularly in the laboratory. I should point out that these E-plates also are available as a 96-well format if you have the SP analyzer. We prefer, like I mentioned, the DP analyzer because we really want to be able to study both the proliferation and migration. So the E-plates and the SIM plates both have integrated into them a gold microelectronic biosensors, which are going to be able to detect cells cell attachment and cell proliferation through the RTA analyzer. And I will tell you a little bit more of how that process works. So the E-plates and the SIM plates have integrated in them, and I just mentioned these gold microelectronic biosensors. I think you can see the gold in this picture. And by applying a low voltage across each electrode, an electric field is generated. If this electric field is disrupted in any way by either cell attachment, as shown here, or by cell proliferation, or even just by changes in cell morphology, then the control unit will actually have a digital readout that is generated that detects the impedance value. So across this electric field, having the cell attachment will record an impedance value, and that will give you a digital reading. So showing here a different view of that. Here's the electric field across each gold electrode. Then a cell attachment disrupts this electric field. A digital reading of the impedance value is recorded and plotted on the computer that is sitting outside from the incubator, next to the incubator. And then as you have more attachment or larger cells, anything that is going to impede this electric field, you'll have a greater cell index value. And I'm showing you this because I'm about to show you data that looks a lot like this. On the x-axis, we show time elapsed in hours, and on the y-axis, we show you the plotted cell index values. The SIM plate works similarly to the E-plate, except that it consists of a two-part system, an upper chamber, as you can see here, and a lower chamber, which is separated by a microporous membrane, which is depicted here. And the chemotractant that you're testing is typically placed in serum-free media on the bottom chamber. And then the cells that you're interested in looking or studying are going to be seated on the top chamber. And if the chemotractant is going to elicit a migratory response, the cells will attach to the microporous membrane and then migrate through the pores and attach on the bottom side of the microporous membrane, which contains the same microelectrodes that I just mentioned to you are present on the bottom side of the E-plate. So for every cell that migrates to the bottom, it disrupts the electric field that I mentioned before, and then you get a similar digital readout of or a cell index that indicates cell migration. The more cells that migrate to the bottom side, the higher the cell index. And of course, all of these experiments are done in comparison to some kind of control. So, and this is how you really gauge your cell index values as a valuable output. Similar to real-time PCR, you have CT values, and you compare those to your negative controls, and that's how you gauge an actual response. So, in optimizing conditions for migration, some of the things that we really need to pay attention to is the number of cells that are seated at the top chamber. We have found in our hands that 50,000 cells is what works best for us for most of our cell types, mostly because they're primary. If they're not primary cells, usually we can use lower numbers of cells and still achieve really good results. You also have to optimize the concentration or the type of gel layer that is important sometimes for getting the cells to really attach and migrate to the other side. A lot of times it's been recommended for us to coat this microporous membrane with fibronectin, for example. But we've done a fair amount of really looking at the different types of typically extracellular matrix components that might be useful in getting cells to attach and migrate to the other side, including collagen. And of course, the chemotractant is important, right? And the concentration of the chemotractant is going to be critical in really eliciting a good response, a good migratory response. So those three things are really key in getting the migration assays to really function reproducibly in the laboratory. And so just to give you an example of one of these experiments that we've done, this is an experiment using human epidermal keratinocytes. I'm showing you as the schematic here at the top, more or less how we've set up just these two samples. On the left is our negative control, serum-free media on the top chamber with cells, 50,000 cells. And on the bottom chamber is just 0% serum-free media. And so cells should not be coaxed to move to the other side. It takes energy for the cells to really attach and migrate through the pores and move over to the other side. If there is nothing, no serum or anything, any chemoattractant, the cells are really not going to expend energy to move over to the other side. There might be some passive migration, as you see here, with a little small rise in cell index, but it's not a major rise in cell index. In contrast, if we add 10% fetal bovine serum on the bottom side as a chemoattractant as a positive control, you see that there is an increase in the cell index relative to the negative control. But we wanted to see if we could get a better response with a different extracellular matrix component. And so here what we're showing you is a similar result, but this time the microporous membrane has been coated with collagen 1. This is rat tail collagen 1. And again, in serum-free media, you have very little migration, passive migration, but in the presence of serum, you get a much higher response with coating with collagen 1. Similarly, collagen 4 has a similar effect as collagen 1. You get really nice separation of cell migration cell indexes or cell indices. And in doing this, we were able to really get a good sense of what the different extracellular matrix components really look like in terms of being able to facilitate a migratory response with human epidermal keratinocytes using 10% fetal bovine serum as our chemoattractant. So as you might imagine, this is only just one type of chemoattractant that can be used as a positive control for keratinocytes. Other things like epidermal growth factor have been very successful in eliciting a migratory response on these cells. We did similar experiments with other cell types, including human adipose-derived stem cells, endothelial cells, and fibroblasts. And one of the things that we found there is that keratinocytes really prefer to be starved for a shorter period of time, similar to endothelial cells. 4 to 5 hour starvation period prior to a migration assay is really critical. Too much longer than that, the cells start to really, it's detrimental to their health, and they're incapable of migrating too well. But fibroblasts and stem cells seem to really prefer a longer starvation period in order to really migrate to the other side. And that was one of the things that we really discovered, is that your different cell types are going to require different conditions for them to really give you a nice migration result. Fibroblasts and stem cells are a little hardier cells, so they can be starved for a longer period of time before they really are going to be coaxed to move over to the bottom chamber. So one example here of how we would do an experiment in a laboratory is, again, we're doing human epidermal keratinocyte migration. We have looked at cells that were treated with negative pressure and cells that have been untreated. We have then looked at the secreted factors from these cells. So this is conditioned media from these two different therapies, which we place on the bottom chamber as our test sample. And then we ask, do they have a difference, or is there a difference in migration for human epidermal keratinocytes? And what we can say is, indeed, with therapy, we see that we get a higher keratinocyte migration response versus with no therapy. And of course, in red is a negative control, which is just serum-free media, both in the top chamber and in the bottom chamber. We see similar results when, instead of human epidermal keratinocytes, we use microvascular endothelial cells in our chambers, and we see that, again, with therapy, those cells are coaxed to migrate faster than without therapy. And again, we see the same response with human adipose-derived stem cells. We see that there is a greater migratory response, although not as strong and pronounced as with endothelial and keratinocytes. But one of the things that we really appreciate using the xCELLigence that we would have had a difficult time telling with a Boyden chamber assay, which is an endpoint assay, or with the scratch assay, is that there are differences in the profiles and the kinetics by which these different cell types are attracted to migrate. For example, here you can see that the separation begins almost immediately at 3-4 hours between these samples, and it continues that way for throughout the 24-hour study. Endothelial cells are also elicited to respond differently as early as 3 to 4 hours. But for example, keratinocytes don't start to they are not coaxed to migrate until about 10 hours into the assay, and then they do so slowly up until about 16 hours, and it isn't really until after 24 hours that you really see a great separation and a greater response in migration with keratinocytes. And this is the sort of information that is missed when one is doing a standard endpoint assay like the Boyden chamber. Here we have a lot of data, all of it with standard deviation error bars at every 15 minutes when the data was scanned and recorded. And one of the beauties of doing this experiment, of course, is that in the past, whenever we had to do a scratch assay, we were glued to the microscope for a period of time. Here, we can set up an assay on a Friday and then come back on a Monday, and we have data that we can then present to our director, who usually wanted the data yesterday. And so this has been extremely helpful for us in the laboratory. So I'm going to then switch gears a little bit, and this is just one example of how we use the xCELLigence for migration and for looking at how we stimulate cells. I'm going to switch now to the safety component of things, because as you might imagine, let me move back to biocompatibility here. We are a medical device company, and so as such, we are heavily regulated by the FDA. So we have to follow a standard for biocompatibility testing of our materials, which is ISO 10993. And so what we do in the lab is just provide guidance to our engineers and our materials chemists on which materials are safe to proceed with into a final product. Because official biocompatibility testing still needs to be done through an independent certified laboratory. For example, maybe you're familiar with V.A.C. But for product development, for guidance testing, we use the same exact cell line that is used at these other facilities like at V.A.C, the cell line L929, which is a mouse fibroblast cell line. And we use the same exact method as them. We first begin by soaking our test material in complete media at 37 degrees overnight, and then we place that extracted material in the media onto freshly seeded L929 cells for 72 hours. And typically what we did before is we seeded them in these 24-well plates and monitored them by eye, visually, using a microscope. And as I will show you in a moment, of course, that is not a very objective way of really gathering the data, because the cells can be rounded and detached at various percentages, and it's difficult to really give that a value or a number for cytotoxicity. So what we decided to do was place the cells side by side in xCELLigence e-plates, and then compare our findings. So again, just to recap exactly on how the assay works, L929 cells are seeded freshly 1 day or two before the assay. In the meantime, the material that you're trying to test is going to be extracted in complete media. Then that complete media is going to be transferred onto the wells containing these cells. And if there's a cytotoxic component in that media, it will have leached out. I mean, in that material, it will have leached out into the media, and so the cells will be become eventually very irritated, and then they will die. We monitor for 24, 48, and 72 hours. And as you can see, what the guidance tells you is that you have a score, a reactivity, and this is the guideline. No more than 20% of cells are loosely attached, no more than 50%, 70%, etc. Well, it gets really tricky at some point to really give this a percentage if you're doing it by eye. And anything that is higher than a 2 fails by the FDA's guidelines. So it's really important to be able to accurately tell whether something is just a mild irritant or a moderate irritant, because it might really preclude you from being able to move forward with that material. And in many cases, when I was doing this in the laboratory with my coworkers, I would give something, depending on my mood, a 2, and while the other person might give it a 3, and it was very difficult for us to really match our reactivity findings. And so we decided for that reason we wanted a more subjective test, and we started using the xCELLigence side by side with our own visual experiment. And of course, we sent all of these samples out later on to an independent certified laboratory to confirm the findings. And what we found from more than 10 different times that we did this was that the xCELLigence always matched the GLP certified laboratory results, whereas sometimes our eye was a little bit less well trained and would sometimes maybe give something a failure score when it could really just pass. And so from then on, really, we've been using the xCELLigence exclusively for getting a biocompatibility reactivity score in order for us to really be certain that we're not scoring it too harshly or too leniently in some cases. So to give you an idea of how the xCELLigence cytotoxicity assay looks, and I think that Lena explained earlier that there are really over 500 peer-reviewed articles from different laboratories that have used the xCELLigence for various purposes, and several of those published articles really detail how cytotoxicity is tested. What's novel about what we were doing in the laboratory is just simply the application. Cytotoxicity has been well established using the xCELLigence, but using it in the medical device industry has been somewhat of a novel thing for us, and it's really been a time saver in a lot of ways. So this is what you typically see in terms of a cell index. You find we see the cells, as I mentioned, 24 hours prior to the experiment, and we see a high number of cells. So you immediately get a high peak in cell index, which then plateaus for the 24-hour period because it is a really highly confluent well. At that point, we add our material, our extracted material, which, if it's a positive control, would be cadmium chloride. And cadmium chloride is a really highly toxic compound. And when we do this, for example, as a control, we immediately get a change in morphology, a hump here, and we see a really quick descent in the cell index to really rapidly approach a cell index of 0 again. And that's because the cells have rounded up and then they have detached. So now these electric fields are not disrupted. So you get 0 impedance, and so the cell index is 0. When, of course, the cells are still attached, like with a non-toxic extract or a negative control, which is just media alone, then, of course, the cells remain attached. We have disrupted this electric field, and so the cell index remains at greater than 1 for the duration of the experiment, which is typically the 72 hours post-addition of the extracted material. And so this is typically how we do a cell biocompatibility assay. Sometimes some materials will give you an intermediate response. And so what we do is we calculate numerically how the percentage from the negative control and the positive control. And based on that percentage, we're able to calculate or assign it a reactivity score of 2 or 3 or 4. And we'll talk about that in a second. So just to give you a sense of how the data looks again with other controls, in green, we have just a negative control that's been extracted in media. In fuchsia, we have a negative control, which is just media alone. In blue, we have a butadiene O-ring, which is an intermediate cytotoxic control. And then, of course, we have cadmium chloride, which is a positive control for death. On the right-hand side, we also did these experiments, as I mentioned earlier, side by side on 24 well plates. And this is what they look like. You can see the cells pretty much look the same for the negative controls. For the buna, you can see a pretty nice decrease in cell number. And with cadmium chloride, you can definitely tell by eye that you're seeing some cell death. The cells are granular, and this would definitely be scored as a 3 or 4. So this is an example of when things are very easy to tell based on by eye what they're really doing. But there are cases when it's really a little bit more tricky to really see these effects by eye. Of course, this is just the same results at 48 hours, comparing by eye, and then at 72 hours. We wanted to be certain that the data that we were seeing by eye was also going to be relatively the same as with other traditional methods for assaying cell proliferation, which is the standard MTT assay. We use a modification of that, which is WST. And so we cultured the cells. We added our extracted materials, cadmium chloride, buna ring, the PET membrane negative control, and the media-only control. And the higher, this is a colorimetric assay, the higher the proliferation, the more the cells that are present in the well, the higher the absorbance value. And so you can see that with cadmium chloride, as we might expect, we have fewer cells in our wells after 24 hours, 48 hours, and 72 hours. With bunadiene, we have a slightly more intermediate result, similar to what we see with the xCELLigence and by eye. And of course, with our negative controls, we see that the cells are still healthy and viable and showing good metabolic function. So we validated both by eye with the xCELLigence as well as with MTT-WST to ensure that these cell indices were consistently telling us the right story before sending things out to our independent certified laboratories for testing. So here's an example of when things don't exactly look straightforward, and it's a little harder to tell what the cell index should be. At the top is your negative control, the PET membrane, 2 test samples, A and B, and then cadmium chloride. we can see a clear difference between the top sample, it's very healthy and alive, and the bottom sample, which is really starting to die even at 24 hours. And we can see that at 24 hours on the cellular index, here's cadmium chloride, here's the PET membrane, but we're starting to see that A and B are doing something strange. They're really both looking like they're intermediate in their reactivity on the cells. And that could pretty much look like these are reacting similarly to that halfway, an intermediate response. If we stain the cells to get a better sense of what they look like at 72 hours, which is here on the xCELLigence, just a second, sorry, which is really here. This is the PET membrane. We see a lot of cell nuclei, green cells, and there's quite a bit of them. Cadmium chloride, and unfortunately, we see a lot fewer cells, or fortunately, cells have died, and so we see a lot fewer of them. And A and B, we see that on some plates, it's really difficult to tell. You've got a lot of dead cells and some green cells. In other wells, you might have some more green cells and a few red cells, and it's not very consistent or reproducible from well to well. But what we did do is we sent these samples out to the certified laboratory, and they came back with the result that we saw with the xCELLigence, which is at 72 hours, this one in fuchsia was scored as not cytotoxic. Whereas by eye, we really struggled with scoring this result here. So, and this was consistent from many times that we sent out our samples to Aptec. We got better results or more accurate results using the xCELLigence and trusting the xCELLigence values rather than really trying to gauge a score of a 1, 2, 3, or 4 by eye using the microscope. And I should mention here that this is, and I will thank the team later, but this beautiful fluorescence imaging is compliments of Marissa Schmidt in the laboratory. So one more thing that I'd like to just kind of go into when doing these biocompatibility tests, I think you can notice here that these dark circles are the microelectrodes under using microscopy. So we only have a small window where we can actually see and photograph the cells using the standard E-plate. And so I think one of the beautiful things about ACEA and its products is that they've designed the product slightly differently. You have a couple of options. If you want to be able to image the cells and still have a cell index reading, they have now included a new product, which is the E-plate View, which really has just removed, I think it's something like 4 of these microelectrode arrays, and allows you a cell viewing window. So we can actually get a cell index in a 24 hours, still take this and take a nice image between this window using microscopy so that we can really validate the data more readily compared to the E-plate, standard E-plate, which does not have this viewing window. And of course, we can only gauge cell growth between this small area. But we wanted to validate the E-view plates compared to the E16 plates to make sure that the cell index was not being impacted negatively by having removed these 4 different microelectrode arrays. Again, these are controls, negative controls, media, the PET membrane, butadiene, and cadmium chloride. And this is the E-view plate, so you can really see the cells more clearly. And this is using an E-plate and trying to do some microscopy. All of the samples were read by the xCELLigence at 24 hours, 48 hours, and 72 hours. And what you can see is that it doesn't matter which plate you use, E-view or E-plate, you get the same cell index value. So there is no negative response to having these 4 different microelectrodes removed from the plates, which I'm sure ACEA had already validated on their own before launching the product. But it's always something that is nice to repeat in the laboratory to be absolutely certain that our results weren't somehow impacted by the change. And so it's really easy for us to now be able to do some microscopy at the different time points if we wanted to, at the same time that we can get a nice cell index and get a really nice subjective result. And so as I mentioned earlier, the way we do this is our negative control. So we normalize the cell index and give it a value of a 1 and a cytotoxicity score of 0. Our positive control, which is cadmium chloride, we normalize to a 0 and cell index of 0, and of course the cytotoxicity score is a 4. And so anything in between would be in this range, 0.75, 0.5, or 0.25. And depending on where that range is, it will get a cytotoxicity score of 1, 2, or 3. So for samples A and B, for example, where the cell index was 0.14, the cytotoxicity score was 3.5 because it would be here. Whereas sample B, which is a 0.92, the cytotoxicity score would be closer to 0, between 0 and 1. And so this is now how we score our biocompatibility samples. And it really is a lot faster way of being able to tell our engineers and materials chemists that they're on the right track with whatever product or materials are selecting. Because if they were to do this officially through the GLP certified laboratories, doing an experiment like this would really take about a month for them to come back with results. Here we can set up an experiment. We usually have cells growing in the incubator. We can set up an experiment a day later at the extracted materials, and then 3 days later have a result. So in 4 days, we can turn around and tell our engineers that they're on the right track with the material, or let them know that they're going to have some difficulty moving forward with that process, instead of having to wait month after month in case we have a lot of failures. And so again, previously for biocompatibility, the way we had to score was slight, mild, moderate, severe was really based on a percentage of cells that were still attached at the bottom of our plates, which was extremely difficult to gauge. Now we've changed it so that we can say slight is a normalized cell index of 25% less than cell control. And this value is easy to obtain, and it's a number that you can calculate based on the cell index. So these are the types of experiments that we do currently at KCI. And I just wanted to kind of give you a plug for KCI here, because KCI does an enormous amount of work really to learn more about how to heal hard-to-heal wounds. And heaven forbid that you or a loved one should ever have a hard-to-heal wound, because they really are detrimental to your quality of life. But if ever you are in that situation, please just ask your doctor about some of the KCI products, because they really are great for healing these wounds. But really, we're here to talk about the xCELLigence, and I hope that I've been able to demonstrate in some ways how important the xCELLigence has been for us. It's been an instrumental resource in the laboratory, because we are asked to put out so much data, and there are really only 4 of us in the tissue culture group. And so to be able to get data in overnight, in the case of the migration assays, or in 4 days in the case of biocompatibility, using the xCELLigence with standard deviation and error bars for every 15-minute scan that we do, has just been invaluable for us. It's really helped us to really move forward very quickly in the laboratory. And of course, it makes all of our senior leadership very happy to get very timely results. And so with that, I would like to just say thank you, of course, to the tissue culture group, Leslie, Iris, Marissa, and our senior manager, Tony Reisers, and not shown here, but our previous director, who's just recently left us, Amy McNulty, who had the vision to really give us a go-ahead on really purchasing all of these wonderful xCELLigence instruments. Of course, I'd like to thank the ACEA team today for giving us the opportunity to be able to speak to you about our work and share some of the work that we're doing at KCI with you. And as we mentioned earlier, if you have any questions at all about how, some of the current studies, ongoing studies, even just questions about some of our products, please feel free to contact me at my email address, or visit us at kci1.com. And with that, I'm happy to answer any